Hello, Dr. Melillo. I will be teaching the process of cell division or mitosis. The cell cycle is a series of events that will occur in a cell leading to the production of the two daughter cells. Cells will undergo mitosis when tissue repair is needed because it has been damaged or for the growth of an organism. The first phase of mitosis is interphase. The cell will spend most of its time here, about 90% of its time, because it is preparing for cell division. It is split into four phases: the G1, the S, the G2, and the M phase, or mitosis. The first phase of interphase is the G1 phase. This phase is very important because it decides whether the cell will either continue mitosis and duplicate or go into the dormant state, which is the G0 phase, which we will discuss later. Here, the duplication of organelles occurs, as well as the synthesis of mRNA and proteins, which will be needed for DNA synthesis later. The cell will also increase in size, and when it is finished doing so, it will move into the S phase. This phase usually lasts 8 or more hours, but it varies depending on the type of cell. However, if the cell is either chosen to remain undivided or the required nutrients to duplicate are not available, it will move into the G0 phase. Here, the cell is performing regular cell function, and it is not preparing for cell division anymore. In humans, there are cells that will remain in this phase, for example, neurons, which will remain here their whole lifetime. However, stem cells, which duplicate constantly and rapidly, skip this phase. The second phase of interphase is the S phase. Okay, so when the cell begins the G1 phase, it has 23 pairs of chromosomes, which are tightly coiled DNA strands that will become visible in the nucleus when the cell prepares for mitosis. DNA is what holds an organism's genetic information, and it stands for deoxyribonucleic acid. It is composed of a sugar, deoxyribose, plus a nucleotide base plus a phosphate. In the S phase, these chromosomes will duplicate, and there is a synthesis of proteins in the nucleus. Humans have 46 pairs of chromosomes, and it is very important for this to be precise, because if there is one less or one more, disease can occur, such as Down syndrome. This step will usually last 6 to 8 hours. At the end, each chromosome will have been duplicated, and each chromosome will have two sister chromatids. A sister chromatid is the result of a single chromosome being replicated into two copies of itself. Even though the chromosomes have been replicated, they will not be individually distinguished because they are still in chromatin form. Chromatin form is when it's loosely packed fibers that are useful for copying and protein synthesis. The third phase is the G2 phase, where microtubules will be replicated, specifically the centrioles. Centrioles are cylindrical intracellular organelles composed of groups of microtubules. In mitosis, it organizes the microtubules of the spindle apparatus or spindle fibers. This phase lasts 2 to 5 hours. After this phase, the cell will move into the M phase, or mitosis. Prophase is the first step of mitosis, and it is split into an early and late prophase. In early, astrorays, which are made from the centrioles, will appear and attach to the centromere of each chromosome. So here we have our our cellular membrane, our centrosome, our astrorays, our asters, our spindle fibers, and in the middle are chromosomes and our nuclear envelope. In late prophase, the nuclear envelope will disappear, and the chromatids will connect to its duplicate copy at the centromere. Kinetic cores attach to the spindle fibers, forming chromosomal microtubules. The kinetic cores are protein structures on chromatids where the spindle fibers will attach during cell division to pull the sister chromatids apart. Then the cell will move on to metaphase, the third step of mitosis. I forgot to mention that when the kinetic core attaches to the spindle fibers, the chromosomal microtubules are the ones that reach from the central chromatin to the pole. In the metaphase stage, the spindle fibers will attach to the central to the chromosomes and pull them apart to the opposite poles so they will line up in the middle. The spindle fibers are microtubules that move the chromosomes during cell division. They pull them apart to the opposite poles during metaphase, and they extend from the cell poles towards the metaphase plate. The metaphase plate is an imaginary line that is equal from the two centrosome poles. This is the product of the balance of the pulling power generated by the opposing kinetic core microtubules. During anaphase, the kinetic core microtubules will shorten as well. The last final phase is telophase. Here, the chromosomes will uncoil and both nuclear envelopes will return. A cleavage furrow will also form, which is this. This indent is what will begin cytokinesis, which is the last final state that the cell goes through before it can enter the interphase. Interphase once again. During telophase, the spindle fibers will also disappear. The last step is cytokinesis, where the cleavage furrow will pinch off and form the two new daughter cells. Cytokinesis marks the end of cell division. After this, the cell will return to interphase to perform its regular cell functions.